Welcome to Plantopia. I'm your host, David Goddori, and I'm a plant pathologist at Cornell University. Any listeners who have never met a real live plant pathologist can be forgiven. We are a very low visibility, but very high impact profession. We protect the world's food supply from disease-causing fungi, bacteria, viruses, and nematodes, all of which want to eat your lunch. This time on Plantopia.
We're in a hot spot. I think the hot spot of the United States and maybe the world right now. We've had hundreds and hundreds of students at Iowa State University test positive in the last 2 to 3 weeks. And to somehow think that is a situation we can control without drastic measures is very worrisome.
COVID-19 in the United States has impacted our lives in ways that are beyond the personal experience of most people. Adults who witnessed the 1918 Spanish influenza have long since passed away. Even our societal memory of the COVID-19 epidemic in New York City last spring seems dimmed by a collective amnesia of how destructive the pathogen can be. But tracking, testing, and stopping viruses that would otherwise wreak havoc within a population is the everyday work for some plant pathologists.
We have a lot of experience with testing for very, very large numbers of plants or seeds, for example.
In this episode, we'll explore the parallels between suppression of virus epidemics in human versus plant populations, and how plant epidemiologists deal with many of the same issues as their colleagues in human medicine, such as the need to test millions of individuals and the accuracy and sensitivity of testing. That's today on Plantopia.
Hi, my name is Cristina Fulladolsa. I'm the director of the Plant Diagnostic Clinic at Colorado State University.
My name is David Carlyle. I'm a family physician in Ames, Iowa with the McFarland Clinic. I also am a hospice professional.
So, Cristina, you are working in a diagnostic lab. I assume that a fair number of samples that come across your desk are actually viruses that attack plants. What does that look like? How do you know that a virus is involved?
Well, the first thing that we do is we look at the symptoms that the plants are showing. And virus symptoms can be quite unique. They form patterns either in shadings on the leaves or streaks, or there can be deformations or rings that we look for. That's the first thing that we assess in order to determine if a virus is involved in the disease.
It sounds a bit similar to the process we go through in assessing the risk of someone contracting a virus disease like COVID-19, where the first tier of screening is really questions about, do you have a fever? They will take your temperature. They will ask you questions about where you've been and who you've been with. Have you been in any situations that place you at risk? How does that compare to the diagnosis of virus diseases of plants?
That is actually very similar to what we do. So, for example, if I get a sample that where somebody suspects a virus, and I look at the sample and I think it's consistent with what we would expect viral symptoms to look like, then I'd give the client a call and I'd ask questions about what the spread of the virus has been in their environment. If they see that all the plants are affected or if only some of them are affected. If it's a greenhouse, for example, if the disease has occurred or if the symptomatic plants are closer to the edges or coming from one side, or if they're spread out. And we ask a lot of questions about other things that in the environment that could contribute to the disease, like are there any vectors? Often insects are vectors of viruses, and so we'll ask about that as well. So there are a lot of things that are parallel to what is done for human viral diagnostics.
And then there is the second tier testing that is done, which is much more of a definitive diagnostic test involving molecular methods. How does that work?
So if I get a plant and I suspect a particular virus, what I'll do is I'll look for any tools that might be available, any test kits that might be available in order to detect the virus. And that involves detection of either the protein associated with the virus or nucleic acid detection. Similar to what we're hearing now with COVID-19 about either RNA testing or antibody testing.
The present emphasis in testing for the presence of COVID-19 seems to be geared towards very high volume tests with very high throughput, with very fast results. Do we have anything like that in detection of plant viruses that might inform the methodology used against COVID-19?
We do. We have a lot of experience testing for viruses in plants. Viruses are one of the biggest concerns for trade, so for movement of plants, which is something that I think we can relate to now in this time that we're living with COVID-19. Movement of people, it needs to be restricted. So with plants, we try to limit how much virus is moved when we trade seeds or any plant material that we exchange for growing plants in other regions. To do this, we have set up sampling and testing methods that are high throughput. And a lot of the time, we're looking for something that is not just fast, but that's also reliable, that it's sensitive so that it can detect different levels of virus, and that is not very expensive.
So if I were to tell you that I had a population of around 330 million individuals, and I really wanted to know how many had and had not been exposed to the virus, is this something that would just be too daunting for a plant pathologist?
No, I don't think it's daunting for a plant pathologist unless it's something new. We have a lot of experience with testing for very, very large numbers of plants or seeds, for example. So if you think about how much seed is traded in the world, it is way beyond 330 million. However, we don't test every single seed. So we apply sampling methodologies. And sampling methodologies, it's not a simple task. There's a lot of, we need to understand the epidemiology of the virus, of the viral disease. We need to understand how it's spread and how easily it can be detected, how often it occurs within the population that we're looking at, so that our sampling can be representative of the whole population.
Have you been in touch with your colleagues on the human side of virus diseases to try to sort of compare notes and see where there might be some synergy between the two sciences?
I have personally not been in touch with them, but we use a lot of the literature that is, or the research that's developed by both sides to better understand the particular viruses that we're studying. An example of this is the first virus that was ever studied was tobacco mosaic virus. And from there, we learned a lot about just viruses in general, and it provided information and a base for the study of animal viruses. And so there are many parallels, and how we go about developing diagnostic tools or understanding the biology of these pathogens is similar. And it's important that we continue to share the knowledge of with each other so that we can advance and make more progress.
Hi, David.
Hi, David.
we could just bat this back and forth all day long. John's mentioned a little bit about what this podcast is like, and I'm sure Mark has discussed it with you. But this particular episode we're working on has to do with some parallels between the present coronavirus epidemic and epidemics of plant diseases. So by training, I'm a plant epidemiologist, and I'm struck by just watching the news and hearing my counterparts in human pathology talk of the many parallels between the two. Their discussions of social distancing seem to me very much like multi-line cultivars, a way of distancing susceptible hosts within a population. And I'm just wondering what your perspective is, and having spent some time with Mark, what parallels do you see between human pathology and plant pathology?
The COVID experience since early this year has really made us understand the seriousness of infectious disease. And you go through times and you realize that infectious disease has been really done well with various parts to help protect human beings, and with a novel virus like this. And growing up and living in Iowa, you hear about diseases that go through the plants and go through the trees and wipe out Dutch elm and et cetera, et cetera. I do think there is some similarities between a novel infection going through a population in plants as it is going through human beings today.
Are you familiar with the some of the screening and testing services that are routinely used in plant pathology to test for the presence of these viruses?
I've done a little bit of preparation. In my understanding is that you'll look for DNA sequences that go along with the virus, or RNA sequences that go along with the virus, and you'll look at potential seeds, or you look at different parts of the plants. But I'd say it's all very rudimentary, what my knowledge is.
Yeah, I'm by no means an expert in this field, but what I am struck by is the high throughput of some of the systems that are used for, in particular in the nursery trade, where they are screening thousands, hundreds of thousands, even millions of individuals over longer periods of time for the presence or absence of a particular virus. And I wonder what the potential is for cross-fertilization of these technologies between the need to test extremely large numbers of samples for the presence of a coronavirus in a human population and the routine screening that's done presently within plant populations for viruses across a broad variety of virus families.
I think that human medicine may be able to take some information and some processes from plant pathology, because I would have to think that you're into pool testing, and I'm very, very interested in pool testing for coronavirus. I'm also has a specialization in geriatrics and work with certain nursing homes, and they talk about do pool testing where they would collect samples from like everybody who works at a nursing home like once a month or once a week. and just do the pool test. And if it came back negative, you can say, okay, the population of employees, at this nursing home are fine. Whereas if it turns positive, then you could go back and individually test each and every person who had submitted to the pool test. So I don't know if that goes down the line of thought you're coming up with, but I'm very interested in that element.
It does, because we would typically apply that when we expect the results to be negative. So it doesn't make sense to do individual testing in that situation, because you're expecting to just collect a lot of zeros. You end up with really the same decision matrix if you test 100 individuals pooled, and then you're testing only 1/100th the number of samples. If you get a positive somewhere in that group of 100, then you retest the 100 individually. But it's a much more efficient process, let's say, when you expect the results to be 1 in a million in a positive.
And that's exactly what I'm very interested in congregate living, for nursing homes and for prisons and for other places where you have people who by necessity have to be close to each other, and you want to do, and this is, you can be healthy all the way, in a monastery and never go out. And yet the next day, if you're exposed to somebody, you can get coronavirus. So I'm very interested in the pool element, and maybe plant pathology can give us some ways to deal with this in a logical fashion.
Plantopia is brought to you by the American Phytopathological Society, or APS, to honor the United Nations celebration of 2020 as the International Year of Plant Health. Healthy plants can help us solve world hunger, stabilize the world's climate, protect our forests, and add beauty to our lives.
Conviron is the world leader in controlled environment systems for plant science research. Conviron's reach-in plant growth chambers, walk-in rooms, and Argus control systems provide precise, uniform, and repeatable control of temperature, light, humidity, CO2, and other environmental conditions. Applications include plant growth, entomology, tissue culture, germination, and other research where tight environmental controls are required. Learn more at conviron.com or contact us at info@conviron.com.
Now, back to the show. What would be a reasonable number of tests to run if your resources were unlimited?
If your resources are unlimited, then you want to test as much as possible, because it gives you a higher chance of determining if a virus is there. However, that sometimes gives you diminishing returns. So there's a point at which it's enough. And that will highly depend on the interactions of the virus and its host. So for all viruses, there isn't one single sampling methodology, but it's very specific to the system in which we're sampling. For example, if we were testing tomato seeds, our sampling methods would be different than if we were testing tomato plants, even if it's for the same virus.
So in the diversity of crop plants that are available, it almost seems like every virus where you have to develop a test is novel. So we have a lot of experience in developing tests to things we have never seen before. How long does it take you to develop a diagnostic test for a new virus?
I think that depends a lot on how much information we have. We need to know how stable the component that we're detecting is. So, for example, if we're detecting the RNA of the virus, we want to be able to develop a test that will reliably detect that RNA with or without any mutations. if we're detecting a protein, we want to know that that protein or that particular site on the protein that we're detecting is not going to change, so that the detection method doesn't work anymore. And so we have to test that a lot. We need to validate these tests, which is as many samples as we can. And with collecting samples from different areas, because that they tend to not be exactly the same. The viruses tend to not be exactly the same in different places or in different hosts. And so in order to develop a effective test, we need to make sure that we cover all our bases.
And so the amount of time that you have available to develop and refine the test produces a more durable test. Would that be true?
Yes, that is true. If we are able to, if to test more, we need more time. And so if we are able to validate our test with more samples, then our test will be better. However, when there's something new that we need to detect, we can develop rapid tests. Or I should say, we can rapidly develop tests that we can use. But the reliability of these tests will not be able to be determined until we use it on more samples.
Well, I think that's the other commonality we can say between the plants and humans regarding viruses is testing is critical, and the availability of a test. And Mark and I were talking about this: the specificity and sensitivity of a test is you want to be able to test, but you want to be able to have a valid testing. And I just was telling Mark about a patient of mine who tested positive. We sat her down and had her quarantined for 2 weeks, and I talked to her on the phone, home monitoring. And now she goes in like 5-6 weeks later she's healthy now, and she's going to go in to do convalescent, to have blood drawn for plasma so she can let her plasma be used to help other people. And her antibody test came back negative. And so the question is, was that a false positive test in the first place, and she just did all this for naught, or did she, or did her antibodies not be produced, or did her antibodies, were produced and gone before she got tested, at the plasma center? So, I mean, testing, and the validity of testing is absolutely critical, don't you think, David?
Certainly, and the tests that are employed for most of the plant viruses that I'm aware of are highly selective and very sensitive and quite accurate. How do you strike the balance to come up with a test that provides an acceptable level of precision and accuracy, but it is useful for long enough that it justifies the investment?
Yeah, so there are more sophisticated ways of detecting viruses, and oftentimes we start with those methods. They're too expensive to be used, or too complex, or they might take too long to be able to detect the virus, to be used in large sample cases. But they help us determine the status of that sample. So the importance of that is that we have a database, or we have a biological collection of samples that we know have the virus and others that we know don't have the virus. And so as we develop the tests, we can evaluate their effectiveness against that database and that collection. And when we get to a point where we feel like the detection is good enough, effective enough, sensitive enough to be able to help us manage the problem, then we can decide that we can use that test. It's in plants that usually is partnered with an economic component. So there's value to the crops. There's a certain amount of plants that you can lose. In humans, that's different.
But I mean, that's where you make smaller the world you're interested in investigating. And I go back to congregate living. I mean, nursing homes and other places where people are a necessity. To me, if you had that kind of testing, you could do on some type of a rapid basis. I have a nephew who works at University of Iowa hospitals and research, and he gets tested every Friday to know whether he can go to work on Monday.
Mm-hmm. And at Cornell University, we are tested based upon our level of contact with students. We're tested as often as twice a week.
Really?
But it's a pooled test, and then retesting if it comes back with a positive.
But don't you think that's a smart way to do it? I mean, if you expect a negative, and so you can pool it, and then if you are surprised, then you can go back and do retesting.
I think it's smart, and I think it's also necessary when you look at the consequences of being wrong in not testing. We would essentially shut down. We've been through that. We've essentially shut down since last March, and we're only just reopening now.
When you're dealing with the populations and lay the public, I think how we phrase things and how we frame it in our language is important. And I loved something I heard on NPR within the last couple of weeks. We never should have used the word reopened when we were talking about dealing with the next phase of the virus. The virus is always going to be there and be contagious until we had a vaccine and adequate treatment. And we should have said, we're going to mitigate and we're going to deal with this pandemic until we have an ultimate solution. I think we made a huge mistake by putting in terms like reopened. I think that was totally wrong.
Correct. It's just like we're going back to normal now.
Exactly.
There is no normal.
You should not have. Because I think you created, an idea in the lay public, and you created momentum. We got to push to get there, and you created people who say, "Well, we can go faster or slower." I don't think we ever could have. I think we should have never even said that. We should have just been upfront and say, "This is a virus. It's going to be there. Humans are going to be subject to it until we have an ultimate solution."
It sometimes occurs in plant virus diseases that initially the viruses are quite virulent, but then become attenuated through time. Is that something that we could look forward to, perhaps, with the present coronavirus?
Yeah, I think that there is a equation between contagiousness and virulence. And if a certain virus is so virulent that the individual that receives the virus dies shortly, and they cannot contract it, cannot give it to other individuals. Ebola, and apparently the first SARS was that route. This one seems to have a virulence for some people, but for many people it's not as virulent, and thus it's easy to have it and then to give it to others. And we see mutations on a regular basis based on the scientific studies I've been reading, that it is changing unbelievably. So I think you're going to see change, and you're also going to see variations of both contagiousness and virulence.
Is the trend expected to be consistently downward, Or are there random fluctuations where we might even see outbreaks of a more virulent strain?
I think that it will, the ones we care about that, are going to be mass, contagious. I think they will be downward at some point. I mean, what you have, obviously the other confounding factor will be the vaccines. And as you know, the flu vaccine is dedicated against certain subspecies of the influenza virus. I think that's the way this COVID will be, is down the road we'll have a yearly COVID vaccine that will go against the most common subspecies at that time. And obviously there will be potential for a very virulent element of COVID that is that the vaccines do not take care of. So I think you got both possibilities. But the more contagious, less virulent are the ones that probably will be more likely to spread across the world.
And are we talking about something that's going to occur within a single, say, flu season, or something that's going to occur over a period of years before it's really noticeable to us?
I think that I've been struck by the scientific reading I've been reading about how well this mutates, and they say it mutates on a very, very, very rapid, regular basis. So I think we're going to see the effects of this, very, very shortly. I think vaccines will come out, and they may be effective, for 1 year, but then there'll have to be a variation of that vaccine, year to year. So I think it's going to be very similar to what we have for the influenza virus, based on my understanding. So, getting back to what you asked me about the mutating to a less virulent form, do you see that for these kind of plants, viruses that you're discussing? Is year 5 less virulent than year 1?
It can be. It's not a given. And there are many exceptions to that where viruses have remained destructive. Perhaps because they are not necessarily lethal to the plant, so they may not be selected against, but they might affect that part of the yield in which we're particularly interested. So, of course, there's a pushback from an evolutionary standpoint in a virus that kills its host plant before the plant can reproduce. But it may not do that. And yet it may be very destructive economically. So we have yes, no, and everything in between.
The other quick question is influenza, we now have Tamiflu and treatments for antivirals. I just can't believe there's any availability of an antiviral agent to go against a plant virus, is there?
It's rare. Our response to most plant viruses is to breed resistant varieties, if that is available to us, or to try to remove the virus from a propagation system if a plant is seed grown and it's a seed-borne virus, or from a vegetative propagation system if the plant is reproduced clonally. So that is where the testing comes in, is when you're trying to clean up your foundation material from which everything else is produced, then the testing becomes critical. And the testing is also critical at the end phase in planting to discover and monitor the introduction of certain viruses that may come into a population, spread quickly, and cause a great deal of economic damage.
I'm always interested in how ideas transmit through different scientific communities. We have this really disconnect sometimes nationally on things like masks and how to deal with certain conditions. When a virus occurs in a a plant that it's affecting great havoc, is there pretty much easy, quick uniformity and consensus about what it is and how to deal with it? Or do you see an element of time when there's multiple theories and nobody can agree on it and people go down different paths?
There was a famous case of a plant disease in Florida, citrus canker. It was unfortunate, but it's an illustration of what happens when people lose confidence, unjustifiably perhaps, in the science. And this was a case where probably the top expert in USDA was advising in an eradication program for the disease as it was introduced. It was a bacterial disease. It spread rapidly, and it mostly spread through backyard trees, through infected backyard trees into commercial citrus plantings. It required the eradication of all trees within a certain distance of an infected tree, presumably because the disease was spreading at an undetected level. The amount of disinformation that came out about that eradication campaign, mostly from homeowners and from their legal counsel, delayed the eradication program by several months, if not years, and allowed that disease to become established in the southern United States, and largely led to a dramatic decline in the Florida citrus industry. So that's a good model for what could happen with COVID-19, where the people who are most knowledgeable are speaking about the disease, and yet they're not being listened to because of just, I wouldn't call it fake news, but I would say misinformation or poor quality information.
I think it's a very apt analogy and a very worrisome analogy because, certainly, Iowa, bringing back students. We've had hundreds and hundreds of students at Iowa State University test positive out of 30,000 in the last 2 to 3 weeks. And to somehow think that is a situation we can control without drastic measures, is very worrisome.
For more information about the International Year of Plant Health, visit us at plantopiapodcast.org. Thanks for listening. Our show is produced by John Bryce. Thanks also to Mark Gleason, Jim Bradeen, Laura Isles, and Roshni Karati. I'm your host, David Gadori, and you've been listening to Plantopia.